Hey, thanks so much, Nan. Hi again, everybody. And thanks for the introduction, Tuan. Thanks for the invitation to speak at this great symposium that you've put together this year. And what a contrast to the previous speaker with solar system size interferometers down to literally interferometers we build for you to hold in your hand. Since we are, can you see my demonstration? I'm almost all movies, so we'll see how that goes. Since we do biomedical research, we need to be especially careful about talking about conflict of interest. And we do have some that are listed here. We've spun out a company and have some other financial relationships as well with companies in our field. I'd like to start by acknowledging the really tremendous group of collaborators that we have, highlighted by these people at the bottom of the screen here, who are primarily are both clinical and engineering collaborators. As you'll see during this talk, almost all of what we do is motivated by clinical needs that these people have identified, and not only identified and send us off to find the solutions, but work with us literally every day to reiterate the solutions thousands of times. I'm going to talk mostly about OCT. This is a technology almost 30 years old at this point, so I won't spend too much time on it. Basically, OCT is scanning interferometry for imaging. The first generation of OCT, which is on top here, the method of scanning was actually moving the reference mirror in order to generate a depth resolved map of reflectivity into some sample. And we would then scan the beam laterally across the sample to generate two-dimensional images as illustrated here. This first generation of technology and the history of OCT over those 30 years has basically been a history of going faster and faster. At least that has a lot to do with our part in it. The reference scanning technology was limited by how fast you could literally move a mirror back and forth, so tens to hundreds of hertz. We then moved into the Fourier domain. The next generation of technology, called spectral domain OCT, is also a scanning Michelson interferometer, but we don't scan the reference mirror anymore. What we do is we detect the color spectrum of the light reflected back from the sample, combined back with the reference on an array detector. You can buy line scan cameras that go at thousands or even hundreds of thousands of scans per second, which means we can do live cross-sectional imaging in tissue for the first time. This generation of technology was actually very successful and has become the tremendous clinical success that OCT represents. The third generation of technology, which we really have pushed hard at here at Duke, is also a scanning interferometer, but instead of scanning either the reference delay or the spectrum, what we're actually doing is literally scanning the spectrum of the light source by using a rapidly swept wavelength light laser, and then having a single channel photodiode reference mirror sitting still. With this, we can go hundreds of thousands to even millions of A scans per second, which means we can actually get to real-time volumetric 3D imaging, which we call 4D imaging. And part of this talk is going to be what we can do with that. The technology of OCT, especially in the middle version, the spectral domain version, the cross-sectional images in real time, has been tremendously successful. It's basically taken over as the primary state-of-the-art diagnostic in ophthalmology. Many of you have had OCTs taken, or if you haven't, your parents very likely have. These slides are pretty old at this point, but the market for OCT passed about $1 billion a year about 10 years ago. It's a multi-billion-dollar market now. There's dozens of companies that market these systems. These happen to be intrasurgical OCT systems, of which there's only 3 commercial ones so far. I'll talk more about that in just a minute. But literally tens of millions of patients every year are imaged with this technology. Starting in about 10 years ago. And we had a lot to do with that, but since 10 or 15 years ago, what we've really turned our attention to at Duke primarily has been the next generation of technology. And we're engineers, we're system integrators, and so we like to make things that go fast. And so we've concentrated very, very hard on going from the sort of table, the tens of millions of patients a year that are being imaged right now have to sit at a table, they have to follow instructions, they have to put their head in a certain place and look in a certain direction. And so the benefits of OCT technology as the standard of care in ophthalmology are not available to anybody that's basically non-cooperative. So if you're unconscious, or if you're in bed, or if you're a child, or someone who can't follow instructions, this technology is not available to you. And so we've from the draw of the clinicians that we've worked with, we've actually decided to work hard on those problems. And there's really 2 primary branches that we've done. The first one was imaging in infants and children. Turns out that infants and children have lots of eye problems like adults do. Some of them social related, some of them like shaken baby syndrome that can really only be diagnosed definitively using this technology. And so the first thing we moved to was actually doing handheld-based versions of OCT for bringing the technology to babies, infants, people that are in bed and that can't sit up and follow instructions. What happened, and that happens about 10 years ago or so, and we actually were able to make some fundamental contributions to understanding the development of the eye, both before and after birth. It turns out that your eyeball, your retina, is not done developing when you're born. It actually continues developing after you're born for a couple of months. We were able to actually follow that out in some publications that we published quite a few years ago. Following that, we got very interested in imaging during surgery. So this actually happened to be a handheld probe that we developed. That was actually spun out into a company almost 15 years ago. Well, what we found was that surgeons, not baby imagers, but surgeons would actually bring this into the operating room, push the operating microscope aside, and use it to image their patients during interruptions during surgery, because the surgeons were the primary users of the technology before surgery to plan their surgeries, after surgery to determine if their surgery was successful. And so it was sort of natural for them to want to incorporate it into surgery. And so I'm going to talk about quickly those two things. First of all, the development of handheld probes, which has really become a specialty sort of our group, primarily motivated, frankly, by the clinical draw of our collaborators who are very concerned about these problems, and also by some extremely talented and motivated students that I had that were just really interested in building not super big, but super small, super accurate, complicated optical devices. This system is just a simple scanning confocal microscope that was switchable between doing confocal microscopy on the retina and OCT. It was really very simple optical design, and was basically did it to demonstrate how well you can do using very straightforward optical. And it turned out you could do pretty well. Because it was so small and we could take it into the neonatal intensive care unit, and our physician colleagues could use it on babies, we were actually able to use it to get cellular resolved imaging of individual photoreceptor cells. There's two images here from an infant and a toddler. These are the big scale cross-sectional on-fast images, and then the dashed line here is actually where the OCT is taken along that line. We could actually resolve individual photoreceptor cells as close as 4 degrees away from the fovea. The fovea is where your cone cells are smallest. They're under 4 or 5 microns. They're out here, they're about 6 or 7 microns. But we could resolve them as close as 4 degrees with this instrument right here, and actually contribute to the development of understanding of foveal development in infants, because, for example, one of these retinal layers in here, there's a whole bunch of physiology around what these layers are, is actually present and develops in between, somewhere between 14 and 25 months. So this was just an example of how small we could make an OCT scanner. These same really talented students then decided to do more that's also related to astronomy. If you want to do better than 6 or 7 micron resolution, you need adaptive optics. Adaptive optics, our astronomers that just spoke are very familiar with this, is the key technology for imaging through the atmosphere of the Earth. It's also the key technology for imaging through your cornea if you want to image with a pupil bigger than about 2 microns. It turns out that your pupil can get as big as 7 or 8 millimeters, but the spot size on your retina actually gets worse rather than better as you image through a bigger and bigger pupil because of the aberrations in your cornea. And if you want to correct for that, you need to build in adaptive optics. So these guys were, undissuaded by this. They basically purchased the smallest deformable mirror you could buy. We used MEMS scanner technology and built all of this into a little handheld probe like this that has all of the components that you used to expect from a whole tabletop size adaptive optic imaging system. And we're able to actually image not only in adults, but in infants, in fact, individual cone cells. They're sort of complicated diagnostics of how we tell what size they were, but down to on the order of 2 degrees eccentricity. Here we're about 5 or 6 micron diameter cone cell spacing. That's what these are. These are radial plots of cone spacing in adult eyes as well as in infant eyes as well. Remember, these are squirming infants. And then one more advance in this direction, which is by a current student, Kristen Hagen, who's also very talented. It turns out that the confocal-based images of individual cone cells in your eye are a little bit imperfect. They depend upon the outer segments of your cone cells being intact. And it turns out that if you actually detect the scattered light coming on either side of the confocally detected returning light, you difference the two, you actually get better images, which have been demonstrated to be better at actually identifying individual cone cells. They give better counts compared to histology. And so Kristen actually added the capability in this still handheld little thing, not only to do scanning confocal microscopy with sensorless adaptive optic detection, with a deformable mirror, which corrects from the aberrations of the cornea, but also collects in three separate channels, confocal, left and right, and differentially detects. And so here, this cone cell mosaic you can see down here is actually the most reliable diagnostic of cone cell density that can be done in a system. Okay, let me get back to OCT. And I want to talk about the other main thing that we've been working on for 10 or 15 years related to OCT, which is imaging during surgery. I have these great colleagues that are surgeons over at the Duke Eye Center who we've been working together for 15 years now. They have a really hard job. When you think about doing eye surgery, surgery on your retina, the surgeon has to stand, not sit, stand at a microscope which is suspended over the patients. They are physically constrained to have their eyes in the oculars, their hands on the instruments for hours at a time. It's not a comfortable thing. And then what they're doing is thinking this problem through. So they're looking through the oculars of a microscope down at somebody's retina, which is this blurry thing at the very bottom of this image right here. They have to manipulate these instruments through ports that are placed in the sides of your eye, so that when they actually have to move their, they have to move their hand to the left to make the instrument at the bottom move to the right. They have to do that inversion in their head while they're trying to understand what this image is showing them. They have very limited depth perception from the very limited stereoscopy that they get from the microscope. It's a hard thing. They train for many years to be able to tell what's going on in this image. And so my closest colleague, Dr. Toth, and I have been working for 15 years on this. She was a primary user of OCT before surgery and after surgery, but she's mainly a retinal surgeon. And she said, why can't you give me OCT during surgery? And so we've actually decided to do that. And through several generations of technology, we're able to really bring this to bear. This is using the very fastest version of OCT, the swept source version of OCT, where again, the laser is sweeping very rapidly. In this case, 100 kilohertz, 100,000 times per second. That generates 100,000 depth scans per second, which we then distribute using galvanometer lateral scanners over a raster scan. In this case, we can obviously trade off the number of frames per second of the complete volume with the number of pixels lateral and axial. But this actually accomplishes on the order of gigavoxel per second imaging, which if Elizabeth is still with us, Elizabeth Hillman was talking about why image fast. Well, I think we should actually make a bigger deal, Elizabeth and us, that this is literally the fastest biomedical imaging technology there is. That is, there's no technology I'm aware of in other types of biomedical imaging or other biomedical sensing that pulls information out of living people faster than a gigavoxel per second or so. There's a lot of engineering behind this. I won't spend much more time on it, including in the acquisition, making sure the latency is fast enough. But the result was about 6 or 7 years ago, we actually delivered the first intrasurgical in vivo 4-dimensional OCT, where this was, I'll talk about the latest generation system in just a minute. This is that 100 kilohertz OCT engine built into the surgical microscope hanging from the ceiling of the operating room. We built the OCT visualization into the oculars of the microscope. There was actually an undergraduate student that developed the heads-up display to enable that. So here's a volume of OCT data next to the video view that they can see. That's the clear microscope view of the retina. Here's a cross-section of that volume taken where this frame is located right here. The surgeon doesn't have many appendages left because they're holding scalpels and they're looking through the microscope, but with their left butt, basically, we gave them the ability to control where they're actually looking through this video. They can actually rotate the block of data. They can control where they're looking at the cross section through it. But this technology was actually quite successful. We've actually done hundreds of patients at this point with this technology. This is an example of a retinal surgery. Retinal surgery is primarily grabbing with tweezers scar tissue on the retina and pulling it off very, very carefully. The retina is very delicate. Surgeons have often described it as being about the consistency of wet toilet paper, and it's among the most important tissues to you in your body. You don't want them to mess it up. And so visualizing this sort of operation where you can actually see the tweezers grabbing this tissue is way easier than figuring out what they're doing from this sort of view over here. And that essentially was the whole point. I'm going through a lot of history really quickly here. This was the situation 6 or 7 years ago. Since then, we've actually moved to much faster technology. This is our latest system, which is actually 4 times faster, and that's multi-gigavoxel per second data. Surgeons have moved to heads-up, actually 3D television displays, and so we're now able to display our data on a heads-up 3D television display. And by going faster, we can actually get much more smooth imaging of the data that we're looking at. And so this actually happens to be a circular scan. It turns out that when we start scanning this fast, resonant and galvanometer scanners can't go that fast. So we actually developed a spiral scan in which the mechanical frequencies required of the scanning technologies are much less severe. We can actually isotropically sample over a whole volume like this. And so we actually were held up by COVID. We just delivered this system just before COVID and have just restarted imaging in human patients since then. And here's what it looks like. So now with true real-time volumetric 4D imaging, here's an example of a retinal surgery where this is the tweezers that the surgeon is manipulating. They're actually poking it down through the retina, manipulating the tissues underneath. This one over here, there's a very big thing going on in ophthalmology right now where gene therapies have been shown to have substantial effects on many serious retinal diseases, but these are very early stage gene therapies. They actually cost tens of thousands of dollars per dose. And so delivering it to the right place and making sure they've actually delivered exactly the right volume to the right place becomes critically important. Here's an example of imaging, actually the subretinal injection of a bubble of gene therapy product, which we can obviously visualize much better in volume than you can in cross-section. You have to get the right cross-section if you're going to rely on the cross-section, and that's much better than the en-face simple view through the microscope. And so that's just a quick tour through OCT, modern applications in imaging in microscopy. I advertise robotics in the, oh, I'm sorry, there's one more thing. Well, during COVID, we had to stick at home and figure out something else to do. So this was a great project that this student, Robert Pratt, came up with. We're still asking the surgeon to look at the color microscope view through the oculars and our 4D OCT through our heads-up display. They still have to look 2 places at once. And so we still wondered, isn't there some sort of image fusion that we could do to actually bring this data together and just give them one final great looking volume for them to look at? And this is a great project that Robert's been doing, basically using data fusion. A lot of segmentation is involved to generate these great looking views that we hope to bring to live surgery relatively soon. And finally, the last thing, I promised some robotics. We've actually got quite a bit of work going on image-guided robotic surgery to address the fact that these manipulations of the retina that these surgeons are trying to do is actually a little bit beyond the feel which they can feel. They actually can't feel what they're doing. They sort of pretend that they can, but they can't. And the obvious solution there, now that we've got 4D imaging data, is actually roboticize the manipulation as well. And we have great work on that I just don't have time to show. But I want to show one final thing, which is just kind of a fun thing, which is actually getting a robot to chase you down and perform OCT on your eye, whether you like it or not. This is a great project that was done by Mark Dralos and another student, Pablo Ortiz, who's here currently. The idea here is actually to mount an OCT system on a commercial robot arm for automated, maybe even mall-based diagnosis of everybody that walks by, or that at least puts their quarter in the slot. The idea here is that there are 2 3D commercial cameras which are identifying your face, localizing where your face is. They will bring the robot basically close to bear on your face. The robot isn't fast enough to move as fast as you might move around, and so we actually have redundant tracking systems. The robot moves on the order of 100 millisecond scale on millimeter accuracy, and then we've actually got optical scanning. We've actually got a sort of complicated optical scanning in there that can chase you down over a millimeter or so. It's actually shining 4 little spots of light on your eye. The Purkinje reflections from those spots are solved in order to detect what direction you're looking, where you are, and how far away from the robot that you are. All those 4 things. And the result is an actual robot, automatic robot OCT imaging system that I will demonstrate on my last slide here. This is the system in operation. I think there's audio here. It actually tells your patient what to do.
Stand on the dot. take a step right. Great. Please hold still.
The
Perfect.
green vector there is actually the direction the patient is looking. It knows that. It needs to know that in order to image the desired place on the retina. And then here's the live B scan. Here's the live volumes that are taken. There's still some shakiness of the patient moving back and forth that we do correct in software. But I wanted to end with that because
Now
that's kind of
let's
fun.
move
I'm
on
joking
to
about the
your
chasing you down, but in
left
fact,
eye.
it is
Perfect.
quite good at imaging as long as you're within about a cubic foot volume. And so one could certainly imagine automated measurements of both cooperative patients as well as the baby imaging that I talked about earlier, many applications of this technology. So let me end there. I really want to gratefully acknowledge all of my collaborators and students that are involved in this, those that are represented in red, or those that are here and currently involved in things I talked about today, as well as our funding sources. I'll end there. Thank you.
Thank you so much, Joe. That was an absolutely fantastic talk. It is amazing to see how a technology can go from a lab demonstration to commercial products and commercial robotic projects. So projects and products, just really, really amazing. And that you could be involved in the whole array. So fantastic sort of trip through OCT.
Thank you.